Hello and welcome to your next lesson for Advanced Higher Biology. In this next section of lessons, we are going to be looking at the synthesis and transport of proteins. So up until now, we've considered the size of the proteome and how it gets to be so much larger than the genome. We've also looked at the need for cells to be expressing different proteins at any given time. Moving on, we're going to be looking at how these proteins are actually made, and today that will begin with looking at the need for intracellular membranes in cells, the fact that these are used to make up different cellular organelles, and that those organelles have a role to play within protein synthesis itself. So in order to make sure you know everything you need to know about this, we're going to be focusing around these three main areas, looking at why eukaryotic systems require a system of internal membranes, looking at the location and the purpose of cellular organelles such as the endoplasmic reticulum, lysosomes, Golgi apparatus and vesicles. And lastly, being able to state the difference between rough endoplasmic reticulum and smooth endoplasmic reticulum. Now, all of these organelles and the difference between rough ER and smooth ER, we are going to be coming back to in the next few lessons as well. So if we consider the need for internal membranes of a eukaryotic cell, well, because of their size, eukaryotic cells end up having a very small surface area to volume ratio. The plasma membrane of these cells is therefore too small to be able to carry out all of the vital functions that those membranes would normally be carrying out. If we were to look at this one as our example, our total plasma membrane available for those reactions to take place on gives us a surface area of 5,400 micrometers squared, and that wouldn't be enough. If we are able to split our cell up into individual membranes, that is able to increase our surface area to volume ratio. Now, within the cell, obviously it's not divided into entirely neat blocks, as in this example. In fact, it's split into different compartments or referred to as organelles. And if we can do this, that's going to increase our total surface area for all of that membrane in order for it to carry out its vital functions. And you can see in this example, it has well over doubled our available surface area. So these compartments, these organelles, are surrounded by the same plasma membrane as would surround the entire cell. And really, if you were to look at the surface of a cell, you would see one surrounding its plasma membrane, which would be external, compared to the several plasma membranes which are surrounding on compartmentalizing our individual organelles. To look at these organelles in a little bit more detail, we're going to consider a little bit more about cell ultrastructure. So as I've said, we've got our plasma membrane surrounding the cell, and in order to increase that surface area to volume ratio, we have all of these organelles within the cell itself, which are also bounded by that plasma membrane. For now, we're going to be focusing on particular areas which are involved both in the synthesis of protein, but also in the synthesis of the actual components of a membrane itself. To begin with, we're going to focus on the endoplasmic reticulum. So if we're going to look at our cell generally, we're going to consider this part here is the nucleus. And as a result, you can see that the endoplasmic reticulum is actually forming continuous tubes which are going to surround the nuclear membrane itself. So the nuclear membrane is able to sort of separate itself out and become a new organelle, which is this endoplasmic reticulum. So it surrounds the nuclear membrane and also is present throughout some different areas of the cell. Now, this endoplasmic reticulum is referred to as either rough or smooth endoplasmic reticulum, and that depends on whether or not it is associated with any ribosomes. When you can see rough endoplasmic reticulum, it has several little ribosomes dotted around on its surface, and that makes it appear to be rough compared to the smooth appearance of endoplasmic reticulum, which does not have ribosomes on the side of it. Now, the endoplasmic reticulum itself is going to be mostly involved in the synthesis of the parts of the plasma membrane itself. Looking at the Golgi apparatus, It is a series of flattened discs, so it's quite important to remember that when we look at these diagrams of cells, obviously we're looking at them in 2D rather than in 3D. So if you were to consider a 3D model of a cell, imagine these as almost a little bit like a minstrel. If you can think of a galaxy minstrel, that sort of shape. So it's a disc shape, but flat, chocolate button, that sort of thing. And they're stacked up within the cell, several discs at once, occasionally with associated end parts. And these particular apparatus, again, are surrounded by that vital plasma membrane that's going to be carrying out all of the different functions needed. And in particular, when we look at the Golgi apparatus, the function is really protein secretion. So once a protein has been produced by the ribosomes, it's sent to the Golgi apparatus, and the Golgi apparatus sort of package it up and secrete it out to the various parts of the cell. And we're going to be looking at this in quite a bit of detail in the next week or so. We also have to consider lysosomes. So lysosomes, seen here and here in this cell, are involved mostly in carrying hydrolases. And hydrolases are enzymes which are actually involved in digestion of proteins, digestion of lipids, digestion of nucleic acids, and digestion of other carbohydrates. So first of all, it's quite important that lysosomes are surrounded by a membrane in order to keep all of those important enzymes away from everything else in the cell, because obviously if they can be involved in protein digestion, they could be involved in digesting other parts of the cell. So these are surrounded by a membrane, and really in order for the protection of the cell itself, and they remain within the cell until they can be required. Lastly, we have vesicles, and vesicles are small areas bounded by a membrane again, which are involved in transporting proteins and different materials between the membrane compartments. So they're going to take some of the packaged proteins from the Golgi apparatus and deliver it to elsewhere in the cell. They may also be used to take material out of the cell itself. So by now you should have a good idea why eukaryotic cells require a system of internal membranes, really because it allows for compartmentalization of the important parts of the cell organelles, and also it provides an increased surface area for the vital functions performed by the internal membranes to be able to happen. You should also be able to spot an endoplasmic reticulum, lysosomes, Golgi apparatus and vesicles if you're showing them in a diagram, and be able to give the purpose of each of these. However, as I've said, we are going to go into a bit more detail about what each of these is actually doing in order to help produce membrane proteins, and also to be able to produce the other proteins involved in the proteome. Lastly, you should be able to state the difference between rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough being the endoplasmic reticulum which has ribosomes present on it, and smooth being that which is without it. So as I say, we're going to be moving on later on in the week to be looking at how all of these different proteins are actually synthesized, and a little bit more about the role of each one of these organelles in doing that.